So here we have it, transcription. A promoter region, a sequence of DNA, tells proteins exactly where transcription should start in a particular region. And we call this region the transcription start site. And at this point, individual RNA nucleotides are going to be brought in and matched with the DNA, one by one, and this will continue until we reach the end of the gene. So on a molecular level, you really get a feel for the chemistry that's going on here. And so for us to really see what's going on at this point, we need to zoom out a little bit more. So if we take a look at the DNA gene as a whole, from start to stop, this is where transcription started, and this is where transcription ended. This was the full length of the DNA gene that we wanted to transcribe and eventually turn into protein. The RNA copy is actually called an mRNA copy. mRNA means messenger RNA. It turns out that RNA serves many purposes in a cell. And so there are lots of prefixes for RNA that distinguish what it can do. And the most important one for us is this messenger status, because the messenger means that it was a copy made from a DNA gene, and that's eventually going to be turned into protein. We call this our mRNA or our messenger RNA copy. Now you might notice a couple of differences from our DNA gene and our mRNA copy. I've drawn these images to be representative of some distinguishing characteristics of the process. So I want you to tell me, how does this mRNA copy differ compared to the DNA gene that it came from? I'm not going to give you any answers to choose from, just a text box. I'd like for you to organize your thoughts and try to answer this question. Don't worry about getting this right or wrong. Just look at the difference between the gene and the mRNA, and whatever difference you notice, I want you to write it down in the text box here.